with the anticodon, binding to the codon. So it means those are codon. Those are codon. Can you see that each of the tRNA brings an amino acid with it and binds to the different codons? So each of the tRNA has anticodon. The anticodon is the 3 base it has. Now, I want to say some few stuffs before we get started fully. Of course, this is a cell. This is an eukaryotic cell. Because why would I say that? Because it's an eukaryotic cell that has a nucleus. Now, this is the nucleus there. I mean, zoomed with the nuclear pore there. And this space, of course, is the cytoplasm, and this is the cell membrane there. Now, one thing I want to remember is this. The protein is always made in the ribosome. You can remember that from your cell biology. But the instruction with which to make the protein is in the DNA. But something is funny. The DNA is rather too big to pass through this nuclear pore and come down here. So there has to be a way we have to send the information that is on this DNA to this ribosome here in the cytoplasm. Now, let me give you some background here. Like I said earlier on, the protein cannot pass through the nuclear pore because it's too big. For this reason, there has to be a way to find a way of transmitting the information, and that brings us to the stages of protein synthesis. Bear in mind, this is O level, so I'm not going to go in details. The first stage is transcription. So let's go right into it. So protein synthesis takes place in 2 stages. First one is transcription, and the next one is translation. Please stay with me. Now, the first stage is transcription, which involves some things. Now, it is very important to remember the fact that this is a DNA. This is a DNA structure that has the base and the complementary base pairs like that. Now, because DNA is too big, the information on the DNA is going to be copied to a smaller version, and that thing is called, that smaller version is called mRNA. So there's this enzyme called RNA polymerase, which will literally bind to this structure, this DNA, and form this. But before that, some other things will have happened. There's something called helicase, which will split this guy into 2. Like, it's going to literally split this, because there's an hydrogen bond holding that together. But like I said, this is not A level, so I won't go into that detail. But bear in mind that there's a lot of things I'm not saying because it's not A levels. So, but what you want to know is this, for the sake of GCSE O level, is the fact that there is going to be formation of mRNA, which is this guy you're seeing here. So the mRNA will be formed with this one of the strands of the DNA. So in this case here, I'm using this upper strand. This one I'm painting black. That's the one I used to form this. All right. So now, but you probably will take note of something here. Now, normally for DNA, you have A to T, G to C, or C to G, like that, A to T. But one thing you want to remember is this: in RNA has adenine, also has guanine, also has cytosine, but does not have thymine. Rather, what it has is uracil. So that means in RNA, what you expect is going to be A to U, excuse me, G to C. So, if it was with DNA, it's going to be A to T. Excuse me, I didn't do that well. One sec. So, if it was to be RNA, DNA is going to be A to T. But because this is, if it is DNA, I beg your pardon, it's going to be A to T. But because this is RNA, it is A to U, because RNA does not have a T, rather it has a U, uracil. This is very important. And that is what you're seeing here. So we are forming complementary base pair with these. So it's going to be A. So it's normally supposed to be A to T, to be DNA, but it's going to be A to U, C to G, C to G, A to U again, then T to A, like that. So don't forget that you can't have a T in a U in an RNA. It's going to be a U instead of a T. Now, the process of copying these to form mRNA or forming mRNA from the strand of DNA is called transcription. So if you are asked what's transcription, simply say it's the process that involves the copying of the genetic code or the information on DNA to form mRNA. I take that again. Copy of the information from DNA to form mRNA, which means messenger RNA. This is very important. Copying of information on DNA to form mRNA, that is called transcription, and it happens in the nucleus. You can see that obviously here. So this is inside the nucleus. Now, hear this. Students often mistake some words. Now, every 3 base pair in a DNA is called triplets, while every base pair in an RNA, in mRNA, is called codon. So this is a codon. 1, 2, 3. That's a codon. Now, bear this in mind. I want to say this in a very careful way. Don't forget that why we are forming mRNA was because DNA cannot pass through the nuclear pore, so we need to find a way to copy the information. So what is happening here is that whatever can happen here is what this mRNA can do. Basically, it's the same thing. So which means that this triplet is equivalent to an amino acid, which also means that this codon is equivalent to an amino acid, because whatever this triplet can do on the DNA is what this codon can do in mRNA. Hope that makes sense. Just know that the name is just where they are found, but they do the same job, because it's copying. It's like you're sending a message to someone. It's like, or somebody representing you to do what you will have done. So basically here, this triplet is just 3 base pair, or 3 base on the DNA, while codon is 3 bases on mRNA. It has to be clear. Now, so that is what transcription is all about. So this mRNA, this one, will swim out of the nuclear core because it's small, it's just one strand. And that's one thing you want to remember. Sometimes you might be asked to differentiate between mRNA and DNA. You should say that DNA has 2, it's a double helix, it has 2 strands, while mRNA has only 1 strand. That's one of the differences. DNA has thymine, RNA does not have thymine, rather it has uracil instead of that. So there are other differences, really, but that's just what's relevant here now. So this mRNA has left the nucleus, going to where? The ribosome. Now, the ribosome, of course, there's a lot I'm not saying because it's not A level. Please bear this in mind. In case you come across and you're an N level student, we're going to say video on this later, for your own level. Now, when this mRNA gets to the nucleus, there are 2 other RNAs that is necessary to finish protein synthesis in the second stage, which is called translation. So translation involves 2 other RNA, which is rRNA, which is ribosomal RNA, and tRNA, which means transfer RNA. For the sake of GCSE, nobody will really disturb you about the full meaning. Just know tRNA is fine, RNA is fine. All right. Now, when this mRNA arrives in the cytoplasm, goes to the ribosome. The ribosome, of course, have 2 subunits, but I won't go into details on that. So this DNA, this mRNA is going to be sandwiched within the ribosome. Actually, seeing it here in there. Then something happens. In the cytoplasm, you have several types of tRNA that carries different amino acids. So this is an amino acid. This is an amino acid. So this yellow thing you're seeing here is an amino acid. Then this is another amino acid. So this what happens. So this tRNA is going to, it has three base pair. It's not a long chain like you have the likes of mRNA. No, it's just like this is how it looks basically, just the way you are seeing it on the screen here. So it has just 3 base pair with an amino acid attached to it. Now, so what happened is this. So in this mRNA, different tRNA will just come. Let me see if I can do that. I will show you a video on that shortly. An amino acid will just come. This, what's it called? The tRNA will just come and bind. Oh, that didn't go very well. And bind. Let me see if I can move that. Can I? Yeah, I think I can. Yeah, and bind to this like that. Once it binds to that, it forms an amino acid. So this amino acid will detach. This amino acid will detach. Let me bring that closer. So this amino acid will detach once this 3 base pair binds to that. Now, before I go too far, I think I need to say something here. This 3 base pair on tRNA has a name. It is called anticodon. Let me bring that very. It's called anticodon. There, anticodon. So this is anticodon. So 3 base pair on tRNA is called anticodon. 3 base pair on mRNA is called codon. Don't forget that. So it is the anticodon that interprets the codons on the mRNA. So what really happened is that so different tRNA will just come, interpret a section of the codon on the mRNA, and just form an amino acid. Amino acid will be, this amino acid here will be released once it binds to a codon there. Then it leaves. It doesn't bind and stay. It binds to it, release amino acid, and go. The next tRNA will come again, move on to the next codon, interpret it, release its what's it called? Amino acid, like that. So while rRNA is assembling it, like putting it together. So different tRNA will just be coming. So with their with those 3 base pair on this called anticodon, bind to the codon, release an amino acid, bind to the codon, release amino acid, and rRNA will be what? Will be assembling it together. So let me show you some animation to make more sense on that. So of course, don't worry about it. You can see, so this is transcription taking place, really. See transcription taking place, see the mRNA being formed like that. See mRNA being formed. So that thing moving across is called RNA polymerase. You don't need to know that so much here. So these were the things I talked about RNA not having uracil. I mean, not having timing, but having uracil. All right. So this transcription taking place. Now let's go on to this transcription. Like I told you, you can see that this is codon. This is just basically what I've explained. Just pause the video here and get that. So this is transcription, which is just copying of the information. Please walk with me. I'm going somewhere. Just walk with me. Now, from the point where mRNA left the nucleus and goes to the ribosome, translation has begun. So translation is actually that process where, that involves the leaving of mRNA to the nucleus, to the cytoplasm, to the ribosome, then all the tRNA binding, all of that. So basically everything here, you can pause the video here, I've explained, but I want to show you the animation. So this is it. So see a transfer RNA with its anticodon going on to, can you say that? So these are the different tRNA binding with the anticodon, binding to the codon. So it means these are those are codon. So this animation didn't show the whole thing, but what's going to happen is this: you're going to see the mRNA, that amino acid detaching and joining a growing chain. So this will make more sense. Now just walk with me. Oh, good. See this. Can you see anticodon on tRNA bind to the codon, and see what happens? They just keep growing. So each of them will just come. These are example of amino acids, valine, proline, tyrosine, all of them like that. So as it binds to that, the chain will be growing. I think I do have an animation. Beautiful. Look at this. This is interesting. This is interesting. So see, can you see that each of the tRNA brings an amino acid with it and binds to the different codons. So each of the tRNA has anticodon. The anticodon is the 3 base it has. So it picks an amino acid, binds, interprets the codon, and release the amino acid. So at the end of the day, you just have a growing chain of amino acid. That's just how it is basic. So this is translation. So you can see that mRNA, this also is mRNA with codons, 3 base pair. There are codons basically. So each of the codons is being interpreted by the anticodon from the tRNA. Do take note of the fact that tRNA is just, it's not a long chain, it's just having codons, so which are just 3 base pairs, so to say. I hope that made sense. It's a quick overview. I hope I was able to really make it quick, really. So, but the truth is, that's just an overview of what protein synthesis is all about. Don't forget that this is basically just O levels. There's more to it, but for the sake of GCSE, you're very good with this. Very good with this. Thank you for this. For your time, please do feel free to drop your comments and send questions to me if you have to. Thank you so much. See you in the next. Bye-bye.